Hello everyone and welcome to the new session of the lecture series, and today's topic is plant growth and development. So in this lecture we'll do the revision of some of the topic of the plant growth and development. Okay, so previously we were talking about in the previous lecture about the development, and then we were discussing about the conditions required for the growth, and we also discussed about the absolute and relative growth rate. And we also discussed some terms such as differentiation, de-differentiation, and redifferentiation. So today we'll start with the new topics. Okay. So first is the plasticity. Okay. So let's see what is the definition of plasticity. So plasticity is, if you search the English meaning of plasticity, it is the ability to mold. All right. So, or you can say it is the quality. Okay, quality or the ability to mold. Okay, or to change. All right, depending on the conditions. So, plasticity in biology can be defined as the ability of the organism to adjust to the changes or alteration in its surroundings, okay, or variations among its diverse habitat. All right. So simply you can say the ability of the organism to change according to the environment. All right. So this is the simple definition of plasticity. So environment can be the habitat of the organism. Okay. So if there is a change in the habitat, the organism can mold itself. Okay, or it can adjust itself. Okay, and it can form a different form or a different shape. All right. So what does plasticity end up to? So plasticity can give rise to different shape of anything, like different shape of leaf. All right. Or you can say structure, different structure of the leaf. All right. So if we talk about the plants, so what happens? Plant can follow different pathways. All right. In response to environment. Okay. Or different phases of life. All right. And what they will do? They will form different structures. Okay. Depending on the environment or the phases of the life. Okay. So one of the example of plasticity is seen in the plants, okay, such as cotton, coriander, and larkspur. So, okay. So in these plants, what happens? Two kinds of leaves are present. Okay. So heterophyly is seen. Hetero means different kind of plants. Hetero means different. Okay. All right, different kind of leaves. So here you can see in this diagram, this is showing heterophyly in larkspur. Okay, so what is happening is juvenile leaves look like these, and mature leaf, they have a different shape. Okay, there is a change in the shape, or you can say form, as well as the structure. Okay, so this is the example of change in the phase of the life. Okay, so we are talking about two phases. First is the juvenile, second is the mature phase. Okay. Next, another example is seen in buttercup. Okay, so in this, what is happening? Again, two kinds of leaves are seen. Okay. So one of the leaf is produced in air. It looks like this. Okay, it has a larger lamina. All right. And the leaves produced in water are dissected. Okay. They are heavily dissected so that air or water current can pass through them. Alright, if the leaf will look like this, okay, like this, then the leaf can break down due to the force of the water current. So the leaves are dissected, okay, in water so that water can pass through them. Okay, so this you can say is the example of the change in the habitat or environment. Change in the habitat, you can say. One is the terrestrial in the air, other one is in the water. Alright, so different shape, form and structure of the leaf develops according to the environment, okay, or the phases of life. This is known as plasticity. Now let's discuss about the factors controlling development. So we have already seen development. What was development? All the stages, okay, all the stages from germination of the seed, from germination to senescence. Alright, so now there are two kinds of factor. First is intrinsic factor, means internal, and then we have the extrinsic factor. Extrinsic factor are external. Alright, now intrinsic factor are further of two types. First is intracellular and second is intercellular. Now let's talk about the intracellular. Intracellular factors means the genetic factor. Alright, means there is a change at the genetic level or change in the DNA, you can say. Okay, or you can write genes also. Okay, and this information is transferred from the previous generation to the new generation. Means they are permanent. Alright. Now, second are the intercellular factors. Now, intercellular factors can be chemicals such as the plant growth regulators. Okay, and within plant growth regulators, we will talk about the phytohormones, also known as plant hormones. Okay, so this was about the intrinsic factor. Now let's come to the extrinsic factor. Now extrinsic factor can be light, temperature, water, oxygen, nutrition and many more. Alright, we've already seen factors controlling growth. These were also light, temperature, water, oxygen and nutrition. Development also is controlled by these factors. So this is a flowchart showing the factors controlling the development. So you can see development is controlled by intrinsic factor and extrinsic factor. Alright, and intrinsic factor are further divided into intracellular, which was genetic factor. Okay, and then we have intracellular, where we have plant hormones. Okay, now extrinsic factor can be light, temperature, water, oxygen and nutrition. Now let's talk about the plant growth regulators. Okay, now plant growth regulators are also sometimes called plant hormones or phytohormones. Now what are phytohormones? They are the chemicals present in trace amount means present in very less amount. Present in trace amounts that affect the growth and development of the plant. Alright, these are known as phytohormones. These are the chemicals which are required in very small amount. Okay? And what is the amount? They are present in nanogram. Alright? So 5 types of phytohormones are observed in, or they were discovered in, mainly. Okay, mainly they are of 5 types in plants. First are the indole compounds. Okay, indole compounds, what do they have? They have the indole group. They have the indole group as parent nucleus. They have indole group as the parent nucleus, and these are usually heterocyclic compounds. Alright, and then we have the adenine derivative. Now what is adenine? Adenine is a nitrogenous base present in the DNA. Okay, it is a nitrogenous base, we all know. Okay, so here an example of adenine derivative phytohormone is kinetin. So kinetin is a phytohormone which is a adenine derivative. Okay, and kinetin has, okay, consists of this N6 furfuryl amino purine. Okay, this is known as kinetin. And you can also write N6 ring. There is a N6 side ring present in the kinetin. Okay, example of indole compound is indole acetic acid. It consists of an indole group. Now third one is the carotenoid derivative. Example is abscisic acid present in plants. Okay, next one is the terpene derivative. Example is gibberellic acid. Okay, there are a lot of gibberellic acid. Okay, many gibberellic acid has been discovered, but GA3 is the most important one, and okay, and the first discovered gibberellic acid. And then we have the gaseous hormone, okay, which is known as ethylene, C2H4. This is also a volatile hormone. It is gaseous in nature. Now, plant growth regulators, or you can say plant growth hormones. Actually, there is a difference between the regulator and the hormones, but in this chapter, we will be considering plant growth regulators as plant growth hormones only. Okay. So, plant growth regulator is a broad term, okay, which includes lot of chemicals other than hormones also, which can induce the plant growth and development. Okay. So, now plant growth regulators or phytohormones are divided into two groups. Okay. There is group 1 and then we have the group 2. Group 1 consists of all the plant, or it consists of the hormones which are plant growth enhancer. Okay. And then we have in the group 2, we have the plant growth inhibitor. All right. And within plant group, okay, examples are of the plant growth enhancers: auxin, gibberellins, and cytokinin. These are plant growth enhancers. All right. So what does this plant growth promoter or enhancer do? They induce cell division, or they enhance cell division. They cause cell enlargement. All right. They are involved in the pattern formation. Okay. Trophic growth. Tropic growth means movement towards any stimulus. Okay. Movement towards a stimulus. Example is like auxin. Then they are also involved in inducing flowering, inducing fruit formation, and seed formation. Okay, so these are some of the roles of the plant growth promoters. Now, second is the group 2, which includes the plant growth inhibitor hormone. An example of a plant growth inhibitor is abscisic acid. All right, and where this hormone is involved, these hormone, or you can say plant growth inhibitors, okay, are involved in plant responses to wounds. Wounds, you can say any cut, okay, or you can say during herbivory also. Wounds can also be created due to either the cut or a damage, or due to herbivory also means when the animals eat the plants. Okay, so these group 2 hormones are involved in plant responses to wounds and biotic and abiotic stress. All right, biotic stress is due to the presence of pathogens or microorganisms, pathogens, parasite. Abiotic stress are due to the environment. Okay, such as drought, okay, very, or you can say high temperature or very low temperature. Okay, so these plant growth inhibitors are involved in various growth inhibiting activities such as dormancy and abscission. Okay, so they are involved in dormancy such as bud and seed dormancy, bud dormancy, or either seed dormancy, or they are involved in abscission. What does abscission means? Falling of leaves. Alright, this is phenomenon is known as abscission. Now, a hormone known as ethylene. This is the point to remember, that ethylene acts as a growth promoter as well as a growth inhibitor. Okay, so in total we'll study about 5 phytohormones. Okay, out of these, okay, only ethylene is a hormone that can cause growth promotion also and growth inhibition as well. Now let's study about the discovery of the phytohormone first, before studying the function of the hormones. So let's talk about auxin first, how auxin were discovered. So Charles Darwin and his son Francis Darwin, what they did, they were monitoring canary grass coleoptile. Okay, so they were working on the canary grass coleoptile. And what is a coleoptile? Coleoptile is a sheath, okay, present around the or surrounding the shoot tip in monocots. Okay, so they were working on canary grass coleoptile. They were monitoring the growth of this coleoptile, or you can say movement of this coleoptile towards a light source. All right, so what they did, if this was a canary grass coleoptile, they illuminated it with light, and then they measured the movement of this coleoptile towards the light. So what they concluded? They inferred or they concluded that there was a substance present. Okay, they said that there is a substance present in the tip of the canary coleoptiles that helps it to move towards the light. So that there is a substance present in the tip of the shoot, means present in the shoot tip, okay, that helps in the movement of the coleoptile towards the light. Now later F.W. Wendt, he discovered and he identified that substance as auxin. Alright, so auxin was involved in the movement of the canary glass coleoptile towards the light source. Alright, so initially what Darwin and his son said, they said there is a chemical or a signal is present. Where it is present? It is present in the shoot tips, or means it is generated or synthesized in the shoot tips and transported or transferred, okay, to the growth area or the growth zone, okay, and causes the faster growth or more growth in the shady region, as compared to the area in light or the region in light. Okay, so again, if this was the coleoptile light, okay, it was illuminated with light, at this region there was more growth in the shade. Okay, this region is in the shade. Okay, there is more growth here, so that's why the coleoptile bended like this, okay, towards the light, and there was less growth on the illuminated side. Here, less growth. Okay. Now let's talk about the discovery of the hormone gibberellic acid. Okay, gibberellic acid, also known as gibberellin. Okay, so what was observed? That there was a disease known as Bacne disease, okay, which is also known as the foolish seedling disease. Alright, so this disease was seen in rice seedlings, okay, and it was caused by the fungal pathogen known as Gibberella fujikuroi. Okay, this was observed in Japan. Okay, so this disease is known as foolish seedling disease. Why? Because there is an increased or enhanced growth of the internode of the internodal region of the stem. Alright, so if you see a structure of the stem, if this is a stem, these are the nodes. Okay, and what arise from nodes? Leaves can arise, buds can arise, branches, okay, flowers, etc., etc. And this region is the internode present between the node. So there is an increased growth of the internodal region of the stem. Okay, so that's why it is known as foolish seedling disease. Now later what happened, a scientist named E. Kurosawa, okay, what he did, he conducted an experiment in which he applied the sterile filtrates of the fungus. Okay, sterile filtrate of the fungus means it was free from other pathogens. Only the fungus Gibberella fungicori filtrate was present in that extract. So he treated the uninfected rice seedling with this sterile filtrate of the fungus. And what you observed, there was the appearance of the symptoms of the disease. Okay, disease also appeared in the uninfected rice seedling when it was treated with the sterile filtrate of the fungus. Now later, the active substance for this disease was later identified as gibberellic acid. Okay, so later they concluded that gibberellic acid was released by the fungus. Okay, so gibberellic acid was produced by the fungus Gibberella fungicori. That's why the name gibberellic acid also. Okay, and this gibberellic acid leads to the increase in the stem length, you can say, or increase in the internodal region of the stem. Alright. Now next is the discovery of the cytokinin. So cytokinin, so a scientist named Skoog and his colleague conducted an experiment, okay, involving tobacco stem internodal segment. So he was working on the tobacco stem internodal segment, and what you observed, that the callus formation from the tobacco stem internodal segments, okay, occurred when callus was formed when these segments was provided with auxin, means auxin was added, plus, okay, some extract from coconut was also added. Okay, from coconut and some other material also. Okay, it was added and it led to the formation of the callus. Okay, so what they observed, that the callus growth occurred from the internodal tobacco segments when auxin was supplemented with specific nutrients, okay, obtained from the extract of the vascular tissues or yeast extract or coconut milk or DNA. Okay, DNA also contain an hormone which is cytokinin like. Okay, so first cytokinin was also discovered from the herring sperm DNA. Herring sperm DNA. Okay, so then later they found that the additional substances, okay, present in the coconut milk or the DNA or yeast extract, that additional substance triggered the successful proliferation of callus. Okay, so for the formation of the callus, what was required? Auxin was required, but in addition to auxin, all these additional substances, okay, plus the extract was added, and then callus was formed. Okay, later Skoog and Miller identified and crystallized this substance, okay, this extract. Okay, they said this extract contained a compound known as kinetin. Okay, they identified and they crystallized this substance. Okay, and they saw that this substance promotes cytokinesis, means it promotes cell division. Okay, so what is the role of kinetin, you can say, or cytokinesis? Cell division is the main role, and this compound was named as kinetin. Okay, so this was about the discovery of the phytohormones, auxin, gibberellic acid, and cytokinin, which are plant growth enhancers. We'll study about the rest of the hormones, or we'll study about the discovery of the hormones, abscisic acid and ethylene, okay, in the next session. Thank you very much for listening.